Um, Good afternoon, everyone. Welcome to the next class of our medicinal biochemistry workshop. I will be happy to know where we did the last time, till which topic we covered, so that I could continue with further. Okay. Then we continue further. So I hope there is no lagging is happening, because in my previous class, lots of lagging was happening. And sorry for the disturbance of the building noise. There is some build construction is happening in my neighbors, and I can't control it. So there's another video being uploaded for someone. So okay, so PCR was done. Fine. So after the PCR, this is not required. We go with the DNA isolation. Yeah, I hope everyone is there with me now and things are fine. So principle of DNA isolation and purification. So DNA can be isolated from the nucleated cells, and DNA is a giant anion in solution. So what are the sources that we include are the blood, buccal cells, culture cells, bacteria, biopsies, forensic samples, all could be used for the DNA sample. And beyond that, what you can do is, what is happening in the DNA isolation, you lyse your cells first, then you isolate the DNA, then you remove all the impurities, and then you precipitate the DNA with the help of alcohol. So DNA isolation is a routine procedure that is collection of your DNA from your subsequent samples. So there are three basic steps: cell disruption, you can do it with the grinding or sonicating, then remove membrane lipids by adding a detergent, yeah. Then isolation of DNA fragment, removing proteins by adding protease, then precipitating the DNA, usually ice cold ethanol or isopropanol is used. Since DNA is insoluble in these alcohols, it will aggregate together, giving a palette upon centrifugation. So this step removes the alcohol soluble salt. So you have your cell lysis, yeah, you add protein kinase A, then you binding is done, then washing is done with the wash buffer, then elution, you elute your DNA with the elution buffer, and at the end you have a pure genomic DNA. So the basic rules for the lysing of DNA is that the red blood cells with gentle detergent such as Triton X100. Then you wash your cells with hemoglobin, which inhibits friction enzymes and Taq polymerase. Then you work on ice to slow down enzymatic processes. Then you wear gloves to protect your samples from you. And autoclave all your solution and store in the fridge. And then keep the palette and supernatant until you have the DNA that you are looking for. So while lysing your cells, you need NaCl, so which helps to stabilize the DNA and remain as a double helix. We require is the EDTA is required with the help of magnesium ions, which is a cofactor for DNAs which chews up the DNA rapidly. Then anionic detergents, SDS, which disrupt the lipid layers, helps to dissolve membranes and bind positive charges of the chromosomal proteins. It should also include protease like proteinase K, which digests the proteins, incubate the solution at elevated temperature for 4 to 24 hours. In order to get rid of the protein, you make a organic solvent extraction in which you have phenol is to chloroform 24:1 ratio. And the protein at the interfere after centrifugation, you have to add 10,000, have to run for 10,000 RPM at 10 degrees Celsius for 10 minutes. Then precipitating the DNA, so you add 2.5 to 3 volume of ice cold 95% ethanol to the DNA and leave at minus 20 degrees Celsius overnight. Then you centrifuge the sample at 10,000 RPM for 10 minutes at 4 degrees Celsius. Then you wash this DNA pellet, remove excess salts and 70% ethanol and air dry. Then you resuspend in sterile distilled water and then store at 4 degrees Celsius, or for longer term you can store it at the minus 20 degrees Celsius. And quantifying the DNA, in order to quantify DNA, this is the formula that you use. for DNA concentration in microgram per ml as a unit. So you calculate the optical density at 260 nanometer, and then you multiply with the dilution factor and 50 microgram per ml as the key factor, and divided by the 1000. Then you will have three different ranges. If your value is coming around 50, that means it's a double stranded DNA. If it is single stranded, that is a 33 microgram per ml. If it is RNA, then it's a 40 microgram per ml. And also, if you want to check the DNA purity, you check them at with the help of optical density of the ratio between 260 to 280. If the value is coming in between 1.6 to 2.0, that means your DNA is pure. But if it's coming less than 1.6, that means there is a contamination of protein. If it is more than 2.0, there is a contamination of chloroform. You need to repurify your sample. So in summary, the sample for DNA extraction is taken. Then we lyse the cells with elevated temperature or detergent and add enzyme in the salt buffer. Then we remove the cellular proteins, then precipitate the nucleic acid with ethanol, and then check for quantitation and purity measurement of the DNA. So it could be has many aspects: agriculture, development, environmental protection, and ensure food security. It has many aspects. So that was it, the isolation of DNA, and I hope I have shared these lectures with you yesterday or not. No, I haven't shared. Did I share PCR then? Yeah, this introduction of biochemistry, it is shared, and also I think structure of DNA is also shared. So 3 main PPTs for the first day have been shared with you. So I hope my video is working fine. What I'm speaking at the moment, it's going without any lagging. Yeah. Okay, this goes on public. So now I want to show you how the DNA protocol.
Before beginning any protocol, you want to make sure that your workspace has a proper flow to avoid contamination of samples or reagents. This example is set up for a right-handed student. Tips and pipetters are on the left, racks and samples are in the middle, and the discard bucket is on the right. As your hand moves through the flow, it never unnecessarily crosses over the sample again. This video begins after students have spit into the DNA Genotech collection kits, capped the tubes to release the buffer, and mixed the solution by inversion. At this point, sample tubes should not be labeled to preserve student anonymity during the genotyping process. Using the P1000 micropipetter, 500 microliters is transferred from each spit collection tube into a new microcentrifuge tube. When pipetting large volumes, draw up the solution slowly to avoid bubbles or contaminating the micropipetter. Once the solution is transferred, close the spit tube lid and cap the sample. Repeat this process for all remaining samples. to beginning this protocol, the heat block should be dialed to 50 degrees and turned on by flipping the switch. If it's heating, the light will glow red. Place samples in the heat block. This incubation must proceed for at least 90 minutes, but can also be left overnight. Tubes containing 20 microliters of DNA Genotek's proprietary purifying solution, PRECIPITATIng, will be provided. Once the samples have been incubated, you will transfer the entire volume to a tube containing the purifying solution. You may notice that the solution in the tube becomes cloudy. This is normal. Mix the solution with a vortex. To turn on the vortex, press the switch down to touch and turn the knob clockwise to ensure maximum speed. Hold one tube at a time and press the top of the black pad to initiate vibration. Repeat this process for all remaining samples. Embed all samples in ice for 10 minutes. Impurities in the solution will be removed by centrifugation. After flipping the switch in the back of the microcentrifuge, press the open button. Remove the metal lid inside by pulling on the knob. Make sure that the tubes are labeled either with letters or numbers before loading them into the centrifuge. Load samples with hinges facing outward. And balance the tubes across the rotor by ensuring that all tubes have equal volumes. Replace the metal lid and press it until it clicks securely onto the rotor. Close the lid to the microcentrifuge gently. Change the centrifugation time by pressing the arrows to the desired time, in this case 5 minutes. Press the arrows by the speed to navigate to the proper RPM. In this case 14.5 thousand RPM is the max this centrifuge can perform and is close enough to our desired speed. Press the start button. The centrifuge will begin to spin. It is suggested that you watch the centrifuge until it reaches maximum speed to ensure that there are no violent vibrations, which may mean that the tubes are unbalanced or the internal lid has come loose. If this occurs, press the stop button immediately. As the spin is going, obtain 4 new tubes and label them. When the spin is complete, the lid to the centrifuge will pop open. Remove the internal lid and remove the samples gently to avoid disturbing the pellet. Place tubes of the same sample in line with each other to ease the transfer of the solution. After centrifugation, you should be able to see a white pellet that has collected on the bottom part of the tube on the side of the hinge. This pellet contains the impurities in the solution. The DNA is in the supernatant. Using the P1000, transfer 400 microliters of supernatant, which contains the DNA, to the new tube. Place the tip on the opposite side of the tube from the pellet and draw up slowly to avoid disturbing it. Repeat this process for the remaining samples. Precipitate the DNA from solution by adding an equal volume of 100% ethanol to each sample . And mix by inversion 10 times. The solution may become cloudy. This is normal. After incubating at room temperature for 5 minutes, place the samples in the microcentrifuge with the hinges facing outward. Remember to balance your tubes. Spin the samples for 2 minutes. Remove the tubes from the centrifuge gently, being mindful not to disturb the pellet which now contains your DNA. Visually inspect for a pellet at the bottom of the tube on the side of the hinge. A pellet is not always visible, so you can choose to centrifuge again or proceed as normal. In this example, a pellet is only seen in samples C and D, but all samples are carried through the process. Using a P1000, slowly remove and discard approximately 800 microliters of supernatant. Place the tip at the bottom of the tube on the opposite side of the pellet to avoid disturbing it. It is not important at this point that all solution is removed. Just get as much as possible. Expel the tip with the solution inside into the discard bucket. Repeat this process for all remaining samples. Visually inspect the tube to see if this pellet is still present. If it has gone missing, make a note of that. Clean the DNA pellet by adding 250 microliters of 70% ethanol to each sample. Do not mix this solution, as it is imperative that the pellet is not disturbed. Repeat this process for all remaining samples. Place the samples in the centrifuge, being mindful of balance, and spin for 2 minutes. Remove the tubes from the centrifuge carefully. In this step, the pellet still contains the DNA, and it is important to remove as much supernatant as possible without disturbing the pellet. So remove and discard the supernatant using the P200 for precision. This will take 2 or more draws. On the first draw, you can expel the solution into the discard bucket and use the same tip to draw up again, as long as the tip has not touched other surfaces. Repeat this process for the remaining samples. Lay out a few layers of Kim wipes. Open the tube and knock it onto the Kim wipe to remove any large volumes of solution still remaining. Leave the tube open and lay it on its side to evaporate any of the remaining solution from the pellet. Repeat this process for all remaining samples and allow them to air dry for 20 to 30 minutes. If the tube still has spots of solution or if the pellet does not look dry, allow for evaporation to continue. If the pellet looks dry, then resuspend the DNA using water. Add 100 microliters of molecular biology grade water directly onto the pellet side. Cap the tube and mix by vortexing for a few minutes. Visually inspect the tube to ensure that the pellet has disappeared or has gone back into solution. If it has not, then continue to vortex or allow the tube to sit at room temperature before proceeding to subsequent steps. Repeat this for all remaining samples.
So that was the complete protocol of DNA isolation.
Solution by adding an
Okay. So now we continue. PCR was shared with you. So little bit introduction. So I will come within 5 minutes, within 1 minute, and then we'll start. So basically biochemistry, molecular biology or biology that is we are talking today, they are all correspondingly interdependent with each other. So studying the structure and behavior of complex molecules found in the biological material and the way these molecules interact to form cells, tissues and whole organisms. So what is basically biochemistry? It's the application of chemistry with respect to your biological processes at the cellular and molecular level. That's the basic definition behind biochemistry. And the medicinal biochemistry, it's dealing with the something to correlate with the medicinal therapeutics, diagnostic, all the um, molecular medicine perspective and connected towards biochemistry, that is known as medicinal biochemistry. So it is emerged as a distinct discipline around the beginning of the 20th century when scientists started to combine chemistry, physiology and biology in understand how the chemistry of living system is working out. So some principles in biochemistry. So cells are highly organized and constant source of energy is required to maintain the ordered state of our DNA. Yeah. Then living processes contains thousands of chemical pathways. Precise regulation and integration of these pathways are required to maintain life. Then certain important pathways, example glycolysis is found in almost all organisms. Then all organisms use the same type of molecules, that is carbohydrates, proteins, lipids and nucleic acids. And instruction for growth, reproduction and development for each organism is encoded in their DNA. So basic building blocks of life, smallest living unit of our organism. They grow, reproduce, use energy, adapt, respond to the environment. Many cannot be seen with the naked eye. And cell may be an entire organism or it may be one of the billions of cells that make up the organism. So there are basic types of cells, eukaryotic cells and prokaryotic cells. So the prokaryotic cells, they are the nucleoid region that contains the DNA. They have cell membrane and cell wall both. They contain ribosomes without membrane to make proteins in their cytoplasm. On the other hand, they have three basic structures, that is nucleus, cell membrane and cytoplasm with organelles. And water is an essential part of our body. Our body is made up of 60 to 90% and it's used in the most of the body reactions. That's why it's also called as universal solvent. So same way like biomolecules, just like the cells are the building blocks of tissues, likewise molecules are the building blocks of the cells. So animals and plant cells contain approximately 10,000 kinds of molecules and water constitutes around 50 to 95% of cells content by the weight. So ions like sodium, potassium, calcium, may contain for another 1%. So almost all other kinds of biomolecules are organic, CHNOPS, an infinite variety of molecules contain carbon and most biomolecules consider to be derived from the hydrocarbons. So the chemical properties of organic biomolecules are determined by the functional groups. So most biomolecules have more than one. So these biomolecules are structures, the building blocks and they are macromolecules. The building blocks are simple sugars and the macromolecules will be the polysaccharides. If amino acids are the then protein, nucleotide, RNA, DNA, fatty acids, lipids. So if they are breaking from the large big molecule to the small, that will be your catabolic reaction in which your energy is dissipated. If from the small they are making big, the energy is required, then that is your anabolic reaction. So together this is known as metabolism. So your monomers could be linked. So cells link monomers by process of dehydration process, so in which we remove the water. So out of this OH and OH in this sugar group, you will join them together to make a double sugar. So this OH, remove this H from here and then remove this OH from there. Then H2O forms out of it. So breaking down of polymers. So cell breakdowns macromolecules by the process of hydrolysis by addition of adding of water. So if the two molecules are joined together, so you can remove them by addition of water. So like it's a reversible reaction of what we did in linking and breaking. So water added to split a double sugar. So sugars are the most abundant organic molecules that is found in nature, initially synthesized in plants from the complex series of reactions involving photosynthesis, and the basic unit of our sugar is monosaccharides. And the function that they do is that they store energy in the form of starch or glycogen, and they provide energy through metabolism pathways and cycles. So supply carbon for synthesis and other compounds, and form structural components in cells and tissues, and intracellular communications also. Then fatty acids, your lipids, which having two structures, that is polar head and non-polar head. The polar head includes hydrophilic and the non-polar head includes hydrophobic tails. So our monocarboxylic acids contain even number of carbon atoms, and there are two types of they will be saturated and unsaturated. Yeah, the saturated will have single bonds and unsaturated will have the double bonds. And fatty acids are the components of several lipid molecules. Example, lipids are triglycerols, steroids, fat soluble vitamins, and the function is the storage of energy in the form of fat. Membrane structures, insulation, and synthesis of hormones. Steroids, the carbon skeleton of steroid is bent to form four fused rings. So 1, 2, 3, 1, 2, 3, 4. So this is cholesterol, first one, and the cholesterol is the base steroid from which your body produces other steroids like estrogen, testosterones are also steroids. This is testosterone, this is estrogen, and so on. So synthetic anabolic steroids are variants of testosterone. So nucleic acids, they store hereditary information. They contains information for making all the body proteins. There are two types exist, that is DNA and RNA. Yeah. And the amino acids, they are the basic building block of proteins which are having alkyl group, amino group, and carboxylic group. And the center, the carbon that is present, it is the alpha hydrogen carbon. So the building blocks of proteins, alkyl group determines the chemical properties of each amino acid and also determine the how proteins will fold. It basically includes the biological function. And the functions as transport proteins, they are structural proteins, enzymes, antibodies, cell receptors, many functions they have. So proteins as enzymes. So many proteins act as a biological catalyst which helps to fasten up the reaction. So thousands of our body in our body, they are present in our body, which controls the rate of chemical reaction by weakening the bond, thus lowering the amount of activation energy needed for the reaction. So they act as a catalyst. They do not interfere with the equilibrium of the reactions and they could be reusable. So they're playing role in the anabolic reaction, catabolic reaction, metabolic reaction, and yeah, they are pretty prominent. So enzymes, they are the active sites, a cleft or groove in an enzyme that binds the substrate of a reaction. So the nature and the arrangement of amino acids in the active site make it specific for only one type of substrate, which just accept one enantiomer. So this is the egg white with lysosomes, and this is with another peptide into it. So we have understood the basics of biochemistry so far, various basic biomolecules of biochemistry that are present, and the types and function of these biomolecules. Yeah. Okay. Now we go to the next lecture, amino acids. So amino acids and its properties. So we will discuss about an introduction to amino acids, their optical properties, acid base properties, buffer characteristics, physical properties, and importance and functional role. So as we have discussed, these amino acids, they are the building blocks of proteins. The general structure of amino acids includes an alpha amino group, an alpha carboxylate group, and a variable side chain, right? So according to this variable side chain, there will be different properties of the carbon of the amino acid. So amino acid could act as a zwitterine, acting as acid and base both. So as amino acids contains ionizable groups, so they can act as an acid and base in a solution depending on the pH of the solution. So due to this nature of amino acids, they are called as zwitterines or dipolarines. They do not have any charge but behave as acid or bases depending upon the pH changes in the solution. So the biological importance of amino acids are that there are over 200 different amino acids occurs in nature, but only 1/10 of these occurs in the proteins. Yeah, and the biological functions that they perform, like they are the building blocks of peptides and proteins. They act as a precursors. Yeah, many biological important molecules are derivatives of amino acids. For example, tyrosine is a precursor of the hormone thyroxine and the skin pigment melanin, and tyrosine is also precursor of a compound abbreviated as DOPA, that is dihydroxyphenylalanine, and it's act as a neurotransmitter, that is transmission of impulses in the nervous system. Like tryptophan is a precursor of a vitamin named nicotinic acid B3. They are the sources of sulfur derived from the sulfur containing amino acids, and they could be also involved in many metabolic pathways such as gluconeogenesis, where it is involved in the glucose synthesis. So physical properties of amino acids. So amino acids are mainly water soluble, which is explained by its polarity and the presence of the charged groups. So they are soluble thus in polar solvents and do not soluble in the non-polar solvent. Yeah, and they have very high melting point and they reflect high energy needed to break these ionic forces, and they are colorless and crystalline substance and soluble in water, and their melting point is actually above 200 degrees Celsius. And from the optical perspective, they exhibit all amino acids except glycine, which does not have any asymmetric carbon atoms. There could be two types from the optical activity perspective: dextrorotatory, levorotatory. In the dextrorotatory, these are the isomers which has ability to rotate the plane of polarized light to the right, and the levorotatory isomer which has the ability to rotate the plane of polarized light to the left. So both isomers can rotate the plane of polarized light by the same magnitude but in opposite directions. So acid base properties of amino acids. So all amino acids, they contain at least two ionizable groups. That is alpha amino group and alpha carboxylic group. So some contain an additional acidic or basic group in their side chain, which are responsible for the amino acids and acid base behavior. So as a result of their ionizability, the following equilibrium reaction can be seen. So this carboxylic group is there attached to the alkyl group, and then H plus ion is removed. And the amino acid NH2 is NH3 plus is done. So since these reactions are reversible, thus they can be act as acid and base, which explains that they have this amphoteric property of amino acids. And what is zwitterine form? This means the neutral amino acids that are present in the zwitterine forms at the physical pH around 7.4, and the carboxylic will be the unprotonated unprotonated and the amino group will be protonated. So both could be act as acid and base, and they could be, the carboxylic group can remove the proton and the amino group can gain the proton. So in this way, around the pH 7, they could act as a zwitterion form. So amino acids can be present in two configurations, that is L isomer and D isomer, which are mirror images. So which differ in the arrangement of the chemical groups around the alpha carbon atom. So all amino acids in proteins are in the L configuration and L amino acids. So here we can see D and L form. So the D alanine form, this is CCC carbon, and this amino group is present, and this is CCC with the amino group present. So if the amino group is present on the left hand side, this is L-alanine. If it is D-alanine on the right hand side, this is D-alanine. So some chemical properties of amino acids. So amino group reactions due to the amino group, this is called nin-hydrin reaction, which is a powerful oxidizing agent that decarboxylate the amino acids, yielding CO2, NH3 and your aldehyde COH groups. So the reduced nin-hydrin react with the liberated ammonia to form a purple color complexes called Royman purple. Some other colored reactions of amino acids. So amino acids can be identified by specific color, like xanthoprotic test by benzene ring of aromatic amino acids, Milan reaction for the presence of phenolic group amino acids, and sulfur test for the sulfur-hydryl group amino acids, that is nin-hydrin test for alpha amino acids. So reactions could be done because of the carboxylic group. So amino acids with the salt, CON with the bases and esters with alcohol. So if there is a decarboxylation is happening, then amino acids will undergo decarboxylation to produce corresponding amines. And this reaction is quite important from the perspective of living cells, as amines like histamines, tyramine from the amino acids made from the histidine and tyrosine. So we have understood the building blocks of proteins of our body, that is amino acids, and we have discussed its various physical and chemical properties of amino acids with understanding their metabolism also. Yeah. So that's a short lecture about amino acids, what they are, how they are being made. Yes, so now we take to the next step. Structural classification of amino acids. So in this we will discuss various classes of amino acids and how they are different type of structures and their role with respect to amino acids. So amino acids, they are the molecule containing an amine group, a carboxylic acid group, and a side chain that is specific to each amino acids. The key elements of amino acids are carbon, hydrogen, oxygen and nitrogen. So amino acids are bases, structural building units of proteins and other biomolecules, and they are also utilized as an energy source. So there are two types, essential and non-essential amino acids. So there are around 20 total amino acids in human body, and they are needed for the replenished tissue, red blood cells, enzymes and other substances. And there are 9 to 12 can be manufactured by the body, non-essential amino acids, and not obtained from the diet. The remaining 8 to 11 essential amino acids must be obtained from the diet itself. So they are non-essential and essential amino acids. The non-essentials are the one includes alanine, asparagine, aspartic acid, arginine, cytosine, glutamine, glycine, glutamic acid, serine, proline, tyrosine. And the essential includes isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. So that's how we have divided these 20 amino acids into non-essential and essential amino acids. And functional classifications, there could be anabolic and catabolic responses in tissues, pH regulations such as glutamic acids and glutamine, and some amino acids They're playing a big role in the urea cycle and nitrogen management, such as arginine, aspartic acid, and asparagine. Arginine, aspartic acid, and asparagine. So some four required these essential amino acids for proteins and energy. So these are isoleucine, leucine, valine, threonine, histidine, and lysine. And the sulfur containing amino acids are required for methylation and glutathione, yeah, methionine and cysteine. So they are also known for making disulfide bridges within the protein. And the neurotransmitters in the brain that are helping in the precursors, so phenylalanine, tyrosine, and tryptophan. So all the neurodegenerative disorders, Alzheimer, Parkinson, so they come under this category. And precursors to heme, nucleotides and cell membrane, glycine, serine, and alanine. And bone collagen specific amino acids, they are proline. So we can see that how we have characterized all these amino acids according to the various functions in our body. Then according to the chemical structure, so we have talked about the functional structure so far. So they could be, according to the alkyl group, could be non-polar and polar, right? And the non-polar ones are the yellow and the polar ones in the cyan color. And there could be also charged and negative, positive charge and negative charge. That is your lysine, arginine, and histidine. Whereas the negative charge are aspartic acid and glutamic acid. And this is the short symbol like GLI, triple amino acid symbol, also one letter symbol, G, A, V, C, P. So these are the different one. Phenyl with F, tryptophan with W, methionine is M, iso is I, leucine, serine T, tyrosine with Y, asparagine with N, yeah, glutamine with the Q, aspartic acid with the D, and glutamic with the E. This is E, arginine with the R, lysine with the K, KR. So that's how they have give their names. So how they are used in human nutrition? So in our human body, from our diet, the 20 standard amino acids that are used to synthesize proteins and other biomolecules and are or oxidized to urea and carbon dioxide as source of energy. And the oxidation pathway starts with the removal of amino group by transminase, and the amino group is then fed into the urea cycle, and the other product of transamidation is the keto acids and then enters the citric acid cycle. Then glucogenic amino acids can be converted into the glucose through the gluconeogenesis. And some non-protein functions. So many amino acids are used to synthesize other molecules. For example, tryptophan, which is a precursor of neurotransmitter serotonin, and tyrosine, which is a precursor of neurotransmitter dopamine, and glycine, which is a precursor of porphyrins such as heme, arginine, which is a precursor of nitric oxide, and aspartate, glycine, glutamine, which are the precursors of the nucleotides. So we have acquainted with the basics classification of amino acids and the basic structures, and we have discussed its various classifications and various functions in serial order about all the basics amino acids, right? So okay, now next is lipids. So biochemistry of lipids. So lipids, they could be of neutral lipids, which contains your hydrophilic and hydrophobic, that loving and hating group of water attracting groups are exposed outside mainly, and the hating groups to be emerged interior. So the neutral lipids are basically a triglycerides made up of 3 fatty acids linked to the glycerol and have no polar group as a phospholipids too. So they have the hydrophobic tail, but they do not have hydrophilic head at all. These phospholipids, then they are the complex compounds containing phosphoric acids in addition to fatty acids, nitrogenous base and alcohol. So there are two classes of phospholipids, that is glycerophospholipids including glycerol and alcohol, and sphingophospholipids including your sphingosine plus alcohol. And let's talk about glycerophospholipids. So these are the major lipids that occur in the biological membranes, and they consist of glycerol 3 phosphate, which are esterified at its carbon 1 and carbon 2 with fatty acids. And there is a phosphodic acids. These are the simplest phospholipids. They do not occur in a good concentration in the tissue, and the phosphodic acid is an intermediate in the synthesis of triglycerols and phospholipids. And other are the glycerophospholipids containing different nitrogenous bases or other groups may be regarded as a derivatives of the phosphodic acids. So this is how the phosphodic acid group looks like. Here is your phosphate group is there, then 3 carbons. The second carbon alkyl carboxylic group is present with the alkyl group, and then on this carbon again your other carboxylic group is present with the alkyl group, R1, R2, 3 carbon and 1 phosphate group. So that's how the phosphodic acid looks like. Then comes lecithins. These are phosphatidylcholine, that is phosphate plus choline. So they are mostly found in this egg yolk, and it is a phosphodic acids with choline as the base. So they represent the storage form of body choline here. So this is the structure of phosphatidylcholine. So this is your phosphate group with the 3 carbon COO, R alkyl group, COO alkyl group, and under this part on the right hand side to this phosphate group, your cholines are added. Then cephalines, they are phosphodiol ethanolamine groups. So ethylamine are the nitrogenous base present in the cephalines. They are phosphoric acids plus ethanolamine. So in this phosphate group we have added this ethanolamine to this here, this structure. Then there is a phosphodieserine. This includes your phosphoric acid plus serine. So you have your phosphodiol, this the same structure. So we keep adding to the end of your phosphatic group different group, functional groups. So according to them their names changes. So this amino acid serine is present in this group, and they are also found in certain tissues also. Then plasmalogens. So when fatty acids is attached to an ether linkage at the C1 of the glycerol carbon, at the carbon 1 of glycerol in the glycerophospholipids, the resultant compounds is called plasmalogens. And the cardiolipins, they are isolated from the heart muscles, and structurally cardiolipin consists of 2 molecules of phosphodic acids but an additional glycerol through the phosphate groups. So the functions of phospholipids. So structural components of membrane and regulate the membrane permeability. It participates in the absorption of fat from the intestine, and the phospholipids are essential for the synthesis of different lipoproteins and participate in the transport of the lipids. Then comes cholesterol, which is found in animals, and it is called animal sterol. So 1 gram of cholesterol consumes per day in our body, and the functions is they provide integrity to the cell membrane and biosynthesis of the bile acid. Biosynthesis of cholesterol, so the source of energy that is required in them is the ATP, and the precursors that used here are acetate or acetyl coenzyme A. So biosynthesis of cholesterol could be occur in 5 stages. First, synthesis of malonate from acetate, then synthesis of HMG-CoA, then production of isoprenoid unit, then synthesis of squalene, and conversion of squalene to cholesterol. So that was the basics of your biochemistry of lipids here. So here we can take a 10 minutes break, students. So we are continuing since 3 p.m. So let's take a break here, and then we meet at 16:20, and then we continue with the rest of the things. So we are done with this phospholipids, we are done with the amino acids, we are done with the PCR DNA isolation, right? Okay, I have to do something with the enzymes. I will do it. This is done, this is done, this is done. Gel electrophoresis left. We are done with the first part. Yeah. So hormonal, this we will do also maybe tomorrow, and this enzyme part. Okay. So today we will do this gel electrophoresis, right? And we will finish this part with the next lecture as uh, nucleotides maybe. Let me check if it is required to you. This I have discussed already. I have discussed everything about them. I can share to you these slides also. It will be good for your studies. This I have taught you in my first lectures. Students, give your Google reviews on this. It will be good for us. Give your honest views. Okay. Let's take a break here and then we see us around 4:30 बच्चे. Just give your reviews over the edu in meantime in your 10 minutes time. Go now and give your reviews, Google reviews. I don't know from where we can do it actually. I can edit my review. So just give your reviews over the EduFabrica like I just did now. Okay, these are my reviews. You can read them. Okay, students, let's continue the last topic of gel electrophoresis. So I have these DNA and RNA that we have discussed already. That let's see if we have time, we will do it. So in this, this is actually part of MBBT, but this also come as a gel electrophoresis. So remember this snowdrop, that is, it's saying that southern means DNA, northern means RNA, and western means protein. So southern blotting is done for DNA identification, northern for RNA, and western for the protein. So, so this is southern blotting, which is done to detect the DNA fragments and have been size fraction by the gel electrophoresis. And this technique was invented in 1975 by E.M. Southern. So in this technique, we exploit the property of radio level probe with this single stranded DNA. And if you want to detect the presence of specific sequence, you can do it accordingly. So in this procedure, the southern blot DNA from the sample is cleaved into the striction fragments with the striction endonuclease, and the fragments are spread apart by the gel electrophoresis. And the double-stranded helix of each DNA fragment is then denatured into single strands by making the pH of gel basic, and the gel is blotted with a sheet of nitrocellulose and transferring some of the DNA strands to the sheet. So next, a probe consisting of purified single-stranded DNA corresponding to a specific gene is poured over the sheet. So here we can see that this is your DNA run over the electrophoresis, and then you transfer over the nitrocellulose membrane, and then you put over the radioactive labeled nucleic acids, then you recognize with the X-rays. So it could be used to detect many diagnostic diseases, monoclonal leukemia, sickle cell mutations, and many more. So let's see how this is done in practically in the lab, in southern blotting. So there will be no sound for that, but just read the subtitles at the bottom. So that was the main concept, that how practically your PCR, or sorry, southern blotting is done in the lab. So now I will take you the RNA. For the northern blotting, RNA protocol is the same. So you can watch that video in the same streaming within that presentation. So I think it's not big, or it is big. It is big. So you can download these blotting techniques and go through it. So I will switch to northern. Is just everything is same. It will has RNA in it, and then there is a western blotting. So in this western blotting, it's an analytical technique which is used to detect specific proteins in a given sample tissue of homogenate or extract, sometimes referred to as immunoblotting. So in this we check about the protein structure. So it was discovered by the laboratory of George Stark at Stanford, and this technique was given by W. Neil Brunett. His name on the southern blotting. And the principle is that then the protein samples are electrophorized, and on SDS page an electrode transferred onto the nitrocellulose membrane, and with the help of primary and secondary antibody we can detect our protein. So in this technique, first the sample of proteins is separated on the basis of their molecular mass using SDS page or 2D gel electrophoresis. And electrophoresis move the protein from the gel onto the nitrocellulose membrane, where the proteins adhere, and detect the specific protein. Antibody of protein must be available. Then nitrocellulose membrane, it has non-specific sites that can bind proteins, including antibodies, which must be blocked with a non-specific protein solution. And then primary antibodies added into the milk solutions bind to the protein of interest. Then to that the secondary antibodies added, and then with the reporter enzymes such as alkaline phosphatase or secondary, which is linked to the secondary antibody, and with the fluorophore or X-force we can see these protein bands. So here our nitrocellulose membrane having various protein parts, and then primary antibody, then towards the secondary antibody, and then with the X-force or chemiluminescence we can detect a specific protein. So let's see how a Western blotting is done as a technique.
In this video, you will learn how to perform a Western blot. A Western blot can be used to identify specific proteins in a sample and provide information about the protein size and relative abundance in the sample. First, fill a tray with blotting buffer. You will be using this buffer to equilibrate your gel prior to starting the Western blot. Next, remove the gel from the gel cassette using the opening key. Line up the arrows on the opening lever with the 4 arrows on the cassette to open the cassette. After trimming the top and bottom of the gel with a straight edge, equilibrate the gel in the tray with blotting buffer for 15 minutes on a rocking platform. Pre-soak fiber pads in blotting buffer so that they are thoroughly soaked. To make a blotting sandwich, obtain a container large enough to fit the gel holder and add enough blotting buffer until the container is filled approximately 1 centimeter deep. Place the gel holder cassette in the container with the black side down and immersed in the buffer, and the white side up and out of the buffer. Lay one fiber pad flat on the black plastic. Next, wet a piece of blotting paper and place it on top of the pad. Be careful to avoid any bubbles between the pad and the paper, and make certain the buffer covers the paper. Take the gel and carefully place it squarely onto the blotting paper. Again, being careful to avoid any bubbles between the gel and the blotting paper. Next, you will be applying a piece of nitrocellulose membrane. Remove the protective sheet from the membrane and wet the membrane with blotting buffer. Carefully place the membrane squarely on the gel. Avoid moving the membrane once placed on the gel, as proteins will begin to blot immediately. Using a roller, remove any air bubbles between the gel and the membrane. Place a second sheet of wet blotting paper on top of the nitrocellulose membrane. Place a second wet fiber pad on top of the blotting paper. Fold the clear plastic side of the gel holder over the sandwich and clamp it to the black plastic side by sliding over the white clip. This tight fit will squeeze the sandwich together. Insert the gel holder into the inner module. Make certain that the black side of the gel holder is next to the black side of the module. Place the inner module into the electrophoresis chamber. Add a frozen cooling unit and fill the chamber with blotting buffer to the level of the white clip on the gel holder. Place the lid on the electrophoresis chamber. Connect the electrical leads to the power supply, making sure the connections are correct, red to red and black to black. Turn on the power supply and run the blot at 20 volts. If a timer is available, set it for 2.5 hours. When the run is complete, turn off the power supply and disassemble the electrophoresis chamber and remove the inner module. Open the module and place it in a container filled with blotting buffer with the black side down. Starting with the first fiber pad, remove each layer until you reach the nitrocellulose membrane. As you remove the membrane, note that the proteins have been transferred from the gel to the membrane. Note that the kaleidoscope pre-stain standards have been transferred and can be seen on both sides of the membrane. You can also see that there are no longer any proteins on the gel. Immerse the membrane in 25 milliliters of blocking solution and incubate it for 15 minutes at room temperature on a rocking platform. Pour off the blocking solution. Add 10 milliliters of primary antibody. Incubate for 10 to 20 minutes on a rocking platform. The platform should be set at a faster setting to ensure constant coverage of the membrane. Pour off the primary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer, and then discard the wash buffer. Add another 50 milliliters of wash buffer to the membrane and let it wash for 3 minutes on the rocking platform at a medium speed setting. Discard the wash buffer. Add 10 milliliters of secondary antibody and incubate the membrane for 5 to 15 minutes on the rocking platform at a fast speed. Pour off the secondary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and discard the wash buffer. Add an additional 50 milliliters of wash buffer and wash the membrane for 3 minutes on the rocking platform on a medium speed setting. Discard the wash buffer. Add 10 milliliters of substrate. Incubate the membrane with the substrate for 10 to 30 minutes with either manual shaking or on a rocking platform. Watch for the color development. Once the colors have developed, rinse the membrane twice with distilled water and blot dry with a paper towel. Air dry for 3 to 60 minutes and then cover in plastic wrap for storage.
I think we are done for today. Yeah. So we do the rest of the medicinal part of biochemistry. So we have discussed all virtual labs today. We have discussed the basics of biochemistry today. We have discussed the basics of PCR, DNA solution. So we have covered the most of the topics today. So please do comment on these Fabrica Google review. I don't know how can you do it, but this is the place where you can give the reviews. So please give this review over the Google. It will be it's a big one. Cannot share here. So yeah. So thank you very much. That's it for today. Then we see us tomorrow, same time, same place. Till then, take care. Bye bye. See you.